Well, thanks and welcome back after lunch. So we start out this talk with my financial disclosures. I have an equity interest in Decibel Therapeutics, who is a new hearing company that's interested, among other things, in eventually developing treatments for hidden hearing loss, and we'll talk a little bit about that at the end of the lecture. So what I'm going to talk about today is sensory neural hearing loss. As I told you in my earlier lecture, I have spent most of my career looking at animal models of normal hearing animals and animals with various types of acquired sensory neural hearing loss, mostly noise damage, but also aging and ototoxic drugs. And of course, as everybody in this audience knows, sensory neural hearing loss is called sensory neural hearing loss because it involves the sensory cells and the nerve fibers of the inner ear. But when I was starting out, the dogma was that the sensory cells were the most vulnerable cells in the inner ear, and the nerve fibers really were only damaged after the hair cells were damaged. And of course, the punchline of my talk today about hidden hearing loss is that actually in many types of acquired sensory neural hearing loss, we believe that actually neural connections can disappear before the sensory cells. So as I said earlier, and as I think everybody here knows, we've got two fundamentally different kinds of sensory cells here. Where's my pointer gone to? There we go. The inner hair cells shown here in blue and the outer hair cells shown here in red. And as I said this morning, 95% of the auditory nerve fibers contact inner hair cells, and the outer hair cells are their biological amplifiers. So what we're going to be concentrating on today is this synaptic connection between the terminals of the auditory nerve fiber and the inner hair cells. And as I briefly alluded to a moment ago, yes?
Would you mind speaking into the microphone?
Yeah.
You're being overwhelmed.
Yeah, okay, I'm sorry. Yeah, okay. Yeah. Okay, now I can see my pointer on my screen. Okay, good. So the classic view, when I was starting my research, and it's still true, is that if you present noise or ototoxic drugs, you'll damage hair cells if it's loud enough, and the outer hair cells are more vulnerable than the inner hair cells. And so there are many situations where you have selective loss of outer hair cells, and the inner hair cells are still pretty good. And since the outer hair cells are the cochlear amplifier, then loss of outer hair cells is going to cause a threshold shift, and the threshold shift will show up on the audiogram here as a classic noise notch at 4 kilohertz. The punchline of the research that I'm going to talk about today is that if you keep turning the sound pressure level down and indeed get to an exposure that only causes a temporary threshold shift, you can actually see in animals, and I think we have accumulating evidence that it happens in us as well, that before you have any loss of hair cells, you can lose these synaptic connections between auditory nerve fibers and inner hair cells. And what is remarkable about this primary neuronal degeneration is that it doesn't affect the audiogram at all, to a remarkable degree. Obviously, if you lost all the fibers in your auditory nerve, you wouldn't hear anything. But you can lose possibly up to 90% of the fibers in your auditory nerve, so long as it's done fairly evenly distributed across the cochlear spiral, and there'll be absolutely no effect on the audiogram. So that's one of the reasons why it has gotten the nickname of hidden hearing loss. So what we believe happens in noise exposure, and I think we've demonstrated it pretty clearly in animals, is that very quickly, perhaps even during a noise exposure that will ultimately result in complete threshold recovery, you can get degeneration of this peripheral terminal of the auditory nerve fiber where it makes its synapse with the inner hair cell. That's why we call it synaptopathy. Once that synaptic connection has been broken, this particular auditory nerve fiber is going to be completely silent. There'll be no spontaneous action potentials. There'll be no response to sound. And it might as well not be there, except, of course, a cochlear implant could stimulate it. It turns out that what we call the peripheral axon here might survive for a few weeks, but what's interesting and important is that the cell body and the central axon can survive for months, years, or maybe even decades. And of course, that's why cochlear implants work, because in people, you can destroy the entire sensory epithelium, and in many people, a very healthy population of spiral ganglion cell bodies and central axons will remain, and you can electrically activate them with a cochlear implant. It's also exciting in a therapeutic sense that if the hair cells survive, and if a lot of them are disconnected, and if that's an important contribution to hearing impairment in age or in noise damage, then there's good reason to believe that we're going to be able to figure out someday how to get these neurons to send out new axons and reconnect them. When that is, we don't know, but of all of the regenerative fantasies out there, I think this is the easiest one, because you don't require to duplicate any cells, you just require some reconnection, not to minimize its difficulty. So I said that this neural loss has been called hidden hearing loss, because it hides behind an audiogram. It's also called hidden hearing loss because these unmyelinated terminals of the fibers underneath the inner hair cell are very difficult to see in classic pathological material. So this is a cat organ of Cordy that was fixed and prepared the way almost all human autopsy material of the inner ear has been processed over the years, and you can see inner hair cells, and you can see outer hair cells, and you can see lots of morphology, and you can count ganglion cells, but if you look carefully, no matter how carefully you look, in the region under the inner hair cells, you can't see these unmyelinated terminals very well. You can get a hint that there's something there, but they could be completely gone and you wouldn't know it. So we've had to develop special techniques to see them in the light microscope, and I'll talk about that in a minute. But first, let me just show you this experiment here. So this experiment was done in 1955 by Harold Schucknecht, and Harold Schucknecht was the chair of was the chair of the department where I did my training, and because he was the chair, I had to read everything that he had written. And in 1955, he took cats, he behaviorally trained them so that he could do behavioral audiometry, and then he exposed the auditory nerve, and he poked around with a surgical pick, and he destroyed lots of ganglion cells without destroying the hair cells. And you can see in this cat here, more than 50% of the neurons are gone, perfect hair cell population, and absolutely no threshold shift. So he and a bunch of other people showed as early as 1955 that it doesn't take a full complement of neurons to detect a pure tone in quiet. So what do you need a lot of neurons for? Well, the analogy I make is like if you take a picture and downsample it. So I took a picture off the internet and I downsampled it, and it's actually remarkable how much you have to downsample this guy to make him unrecognizable. This is seriously downsampled. But, you know, you're removing information channels from the auditory nerve. There may be and although it's easy to see that this is a picture, and it's easy to see that it's a face, and you may think you know who it is, if you brought back all the information channels, you might be surprised. But that's what hidden hearing loss is all. You're taking away information channels, and it makes it harder to perceive complicated signals. So what I'm going to talk about today is divided into 4 parts. The first part, I'm going to show you how we used immunostaining and antibodies to be able to see synapses in the light microscope, which really allowed us to count them and be very quantitative about what happens in noise damage and aging. And then I'm going to take it to human postmortem material and show you the preliminary analysis we have that suggests that maybe the same things are going on in humans. Then I'm going to talk about a study that we have just finished, a very small pilot study, which we and others are trying to extend to see if we can get electrophysiological evidence for hidden hearing loss in young living humans. And then finally a little bit about neurotrophins and the possibility that we can get spiral ganglion cells to send out new processes and reconnect to hair cells. So how do we see synapses in a way that we can count them? So in this schematic here, I'm showing you an inner hair cell and 3 of the maybe 20 auditory nerve fibers that contact it. So each auditory nerve fiber contacts a single inner hair cell by a single chemical synapse here, which if you look at in the electron microscope looks like this. On the inner hair cell side, there's this electron-dense structure we call a synaptic ribbon, surrounded by a halo of vesicles that contain glutamate and some pre- and postsynaptic membrane thickening. So 30 years ago, when we wanted to count synapses, we had to take serial sections and look in the electron microscope, and it's about 1,000 sections from here to here. And when I was a postdoc, I spent 2 years reconstructing the innervation of 2 adjacent inner hair cells and making the counts. So that was fun, and we learned a lot, but it's not the kind of thing you do casually. So the ability to get lots of data on synaptic counts required being able to tag them in some way so that you could look at them in the light microscope and have them become visible. So a number of years ago, other people showed that they discovered a major protein inside the synaptic ribbon. It's called CTBP2. There was a nice antibody to it, so you can immunostain it and make it turn red. And we knew this was a glutamatergic synapse, and there are lots of antibodies for glutamate receptors. And we chose one, and you can make it turn green, and then you can immunostain a cochlea. And the data that I'm going to show you are from mouse initially. And so here we're looking down on a row of inner hair cells, and the red and the green are the synaptic ribbons and the postsynaptic receptor patches. And so if you turn and look at it from the side, a view more like this, you see all these little red-green puncta right next to each other. If you blow up the power, you can see that every red dot has a green dot and every green dot has a red dot, and they're exactly where they ought to be. And so it's quite convincing that this is a nice way in the confocal microscope that you can very quickly obtain a lot of data. So once you've processed the ear, which doesn't take very long, taking this picture and analyzing it takes 5 minutes. And so in 5 minutes, you can count the innervation of 5 adjacent inner hair cells instead of 2 years for 2 hair cells. So it's a big increase in speed. So if you take a normal mouse, and we have a cochlear frequency map for the mouse, it's a very high frequency animal, and you look at the inner hair cells along the length and you count how many synapses there are per inner hair cell as a function of length. This is what you normally see. In the middle of the cochlea, there are about 20 synapses, 20 auditory nerve fibers per inner hair cell, falling off toward the base and toward the apex. And as we'll see, the human doesn't look very much different from this. In any mammal, kind of in the middle of the region, middle of the cochlea, they're on the order of 20 auditory nerve fibers per inner hair cell. So now for the noise damage studies, and these, as you've heard a little earlier, are all studies I did in collaboration with Sharon Kajawa, the former director of audiology at the Mass Eye and Ear Infirmary and a longtime colleague of mine. So we took mice and we overexpose them to an octave band of noise in the middle of their hearing range, which translates into about 8 to 16 kilohertz. And then 1 day, 3 days, 2 weeks, or 8 weeks later, we measured their threshold shifts by otoacoustic emissions or auditory brainstem responses. And so you can see 1 day, 24 hours post-exposure, there's a 30 or 40 dB threshold shift, depending on which measure you're looking at. But by 2 weeks post-exposure, the thresholds have really come back to normal. So this is a very severe but reversible temporary threshold shift. So in the next slide, we're going to look at suprathreshold responses in 2 frequency regions, one where there was only a little bit of damage and one where there was much more damage, as seen in threshold shift, by both of these metrics. And the take-home message is, if you compare the green, which is the sort of pre-exposure view, and the black, which is the 8-week recovery view, not only were otoacoustic emission thresholds normal, but the suprathreshold amplitudes were normal as well. Whereas with respect to the ABRs, the amplitude, the suprathreshold response, does not recover, even though thresholds are normal. And you can see that here. These are some average ABR amplitudes. The green is what it's supposed to be, and the black, you can see, is reduced to about 50% of the normal amplitude at this suprathreshold level. Of course, wave 1 is the sum responses of the auditory nerve fibers. You may notice that if you look at the later waves, which take you up into the brainstem, there's not as much attenuation, and even it looks like the 5th wave here is actually larger in the exposed animals than in the unexposed animals, and that perhaps gets at the issue of tinnitus that we were talking about earlier. There are certainly signs of central gain enhancement, so to speak, in these ears that have recovered thresholds and attenuated wave 1 amplitudes. So when you look at the histology, when you look at these immunostained cochleas and you count synapses at the bases of inner hair cells, here we're adding a 3rd antibody, myosin, in blue, so that we can see and count the inner hair cells. So here's the quantification. If this is what the synaptic counts should have been, then you can see whether you look immediately after the noise or 24 hours later or 1 week later, there's loss of roughly half the synapses in pretty much the entire basal half of the cochlea. And of course, we've gone out to 2 weeks and 8 weeks and 16 weeks, and it doesn't get any better, it just keeps getting worse. And in fact, most convincingly, if you wait long enough, the spiral ganglion cells start to disappear. And this is a post-exposure time scale here. These are unexposed controls that are showing some age-related loss of ganglion cells, but nowhere near what was seen in these ears with a large temporary threshold shift, where 2 years later, near the end of their lives, they've lost half their spiral ganglion cells. So they lose about half their synapses, but it happens very quickly. They lose about half their ganglion cells, but it happens very slowly. So the idea is you immediately lose synaptic connection. The ganglion cell can survive for a very long time, but ultimately it may die. It certainly appears to in the mouse model. So we wanted to make sure that this wasn't something peculiar to mice. Mice are very special animals. They're a very high-frequency animal. And so we went on to look at guinea pigs and chinchillas and rats, and we've actually very recently also gone on to rhesus monkeys, which obviously gets us closer to humans. And in all of these animal models, we took a very simple approach. We said, let's take a noise band, let's put it in the middle of the hearing range, let's put it on for 2 hours, let's take it up to a sound pressure level that if we went any higher, would cause a permanent threshold shift, but let's keep it at a level where thresholds will recover completely. So it's a very severe but still completely reversible threshold shift, and then look at the histology. And as you can see in all these cases, the black is the normal synaptic count, and the colors are either mean synaptic counts in a big group of animals or synaptic counts in individual animals, and the gray is the noise band that we used. And without having to look hard, and in each of these cases, simply following that algorithm for how to choose your noise exposure produced synaptopathy. So it seems with no hair cell loss and complete or virtually complete threshold recovery, certainly within the 20 dB normal audiometric limits that people talk about in humans. So that suggested to us that this is a very widespread phenomenon. It's sort of fundamental to the mammalian ear, and there's every reason to believe that the same thing would be happening in humans. So my colleague Sharon Kujawa is particularly interested in aging and the interaction between noise and aging. So she went on to not noise expose the mice and just look at them as a function of age. And here she's counting inner hair cells, outer hair cells, and measuring distortion product emission thresholds as a function of age throughout the entire lifespan of the mouse. You can see there's not much inner hair cell loss. There's a slow, late onset outer hair cell loss that mirrors quite remarkably the slow onset threshold shift of otoacoustic emissions, but of course that's as it should be. But what's interesting is that at midlife, sort of right exactly in the middle of their lifespan, when there's less than a 5 dB threshold shift and only a few percent on average loss of outer hair cells, there's already a 25% loss of synapses. So again, in the aging mouse model, as in the noise-exposed mouse model, there's very clear evidence for loss of the synaptic connections before there's significant loss of hair cells or significant threshold shift. Well, so how about in humans? I mean, that is clearly the species that we're most interested in. So for decades, people have been studying human autopsy material. You can't biopsy the human inner ear. You can only get it out at autopsy, and people have to donate their ears to science. And at the Mass Eye and Ear Infirmary, we, starting with Harold Schucknecht decades ago, we've been collecting human temporal bones and analyzing them in various ways. But as I talked about earlier, the ways that they were analyzed wasn't conducive to being able to count synapses. So we wondered whether whether we could take fresh specimens that were obtained by the temporal bone bank and immunostain them and do some of the same kinds of analyses that we had done in the animal material. So to date, we have 17 ears that we have obtained, and for this first part of the study, we decided to concentrate on ones that had no explicit otologic complaints. So they died at the nearby Mass General Hospital. They had indicated that their tissue could be used for science or research. We took out the temporal bones, and if there were no explicit otologic or audiological complaints, we would work them up. So unfortunately, we know nothing about their hearing ability other than that they had never sought out explicit help from otologists or audiologists, as far as we could tell in the medical record. So it turns out that so long as the postmortem time before we can get the ears and get them into fixative is less than 10 or 12 hours, we can do a remarkable amount, can extract a remarkable amount of information. So we drill down the cochlea, we decalcify it, we microdissect it, we immunostain it, we get out all the pieces of the cochlea. So this is a complete human cochlea. We have a cochlear frequency map for the human, so we know where along the cochlear spiral the frequencies are, and these white numbers are the inferred frequency coordinates, and we go in and we take pictures all the way along the cochlear spiral. And what I'm going to show you are nerve fiber counts. So here in this slightly higher power view of a segment of a cochlear turn, we can see the green is a neurofilament stain, which stains all nerve fibers in the ear, and the red is a efferent marker that stains the efferent fibers. So in this region here, which is called the osseospiral lamina, we can see the peripheral axons and the unmyelinated terminals of auditory nerve fibers. Here's the inner hair cell area. Here are the 3 rows of outer hair cells. So let's keep increasing the magnification. So here is the inner hair cell area and the 3 rows of outer hair cells. And here what we're looking at is exactly what we're looking at over here. So these are the unmyelinated terminals of auditory nerve fibers coming up to innervate the inner hair cell in a human ear at the 500 hertz region. And here it is in this same, only slightly older than middle-aged individual at the 4 kilohertz region. I'm going to show you in a second that there's no hair cell loss in either of these cases. So these have almost complete hair cell populations, but you can see the incredible difference in neural density at the 500 hertz region compared to the 4 kilohertz region. So these are 3-dimensional objects, and in the confocal you can focus up and down, and you can create a 3-dimensional image stack, which you can then virtually resection and look at as if you were cutting the fibers in cross-section, and that's what we're doing here, so you can count them. So it's very easy to count peripheral axons. And so here's the 1 kilohertz region, and here's the 8 kilohertz region. And it's very obvious that there's a huge loss of peripheral axons comparing these 2 regions in this particular individual. And we can use that same myosin stain to count inner hair cells, and we can tell how many inner hair cells are there, how many inner hair cells are absent. In some preparations, I don't know if you can see them anywhere outside of the first row, there's some little red dots there too. Those are the synaptic ribbons. So in some of the material, the better fixed material, we can even count synaptic ribbons. Like I said, we can always count hair cells, and so we can get very accurate cytocochleograms, as they're called, showing us the survival of hair cells. We have the cochlear frequency map, so we can know where along the cochlear hearing axis we have hair cells and where they're gone. So here are 4 example cytocochleograms of these, quote, normal aging humans, and I want you to pay particularly attention to this one. This is the oldest one in our sample. It's an 89-year-old, and you can see that out to the 2 kilohertz region, at any rate, in this particular individual, there are 80 or more like 90% of all the hair cells are still there. So there's no hair cell lesion in this individual. But in all of these individuals, we can count peripheral axons all the way up and down the cochlear spiral. And here are some of them. This is most of the numbers that we have to date. So this is peripheral axons per inner hair cell, or synapses per inner hair cell, as a function of cochlear location. These are the very young folks, and you can see the numbers are up 13 or 14 neurons per inner hair cell, and here's the 89-year-old. So it's an N of 1, but it's a pretty spectacular primary neuronal degeneration in an ear that has virtually no significant inner hair cell loss. And these are a bunch of more middle-aged people. So we're accumulating more ears, obviously, and we want to get more that have documented audiology, but it's difficult. But it is possible, we believe, to get a lot more insight into the true state of the innervation in the aging ear by looking between the spiral ganglion and the hair cell, a region that was never really looked at before in more classic pathological material. Okay, so now let's turn to the question of, is it possible to begin to diagnose hidden hearing loss, or to see signs of hidden hearing loss in living subjects? So a couple of years ago, Stéphane Maison and I set out to do that in populations of college-age students. And our hope was, in this first study, to obtain sample groups that within that age range might be at the extremes of noise exposure. We wanted to get a bunch of students who had really abused their ears and a bunch of students who had maybe really protected their ears. And so we went to a bunch of local colleges, but among those we included a college called the Berklee School of Music in Boston, which is for popular music. And we went to the Communication Sciences Department at Northeastern to find people studying to get audiology degrees who had their consciousness raised about protecting their ears. And the idea was to sort of separate them into ear abusers and non-ear abusers, do audiometry, including high-frequency audiometry, do electrophysiology to look for signs of wave 1 reduction, and to do speech and noise tests that were really difficult to try to spread the scores, even among people with normal audiograms. And they had to have normal audiograms to be in the study. So when we were thinking about ABR, we knew, of course, that ABRs are much more variable in human subjects than they are in genetically identical mice. And so we decided to adopt a strategy that others before us have used, which is to normalize waves within one waveform to each other. And we were particularly interested in looking at SP, which, as you know, is thought to be a hair cell-derived potential that you can see quite well if you get your ABR electrodes really close to the eardrum, as you can with insert tiptrodes. These are mouse data, and you can see why it inspired us, because in our synaptopathic mice, when we got our electrodes really close to the ear canal, we could see a very prominent SP, and as you can see, and as is consistent with the hair cells being fine and the neurons being disconnected, wave 1 is significantly reduced, but SP is identical. So the idea was, what if you took the ratio between wave 1 and SP? We're also not the first people to think about doing that. That's an old idea. But it looked from our mouse work like if you took the ratio between wave 1 and SP, you might normalize out differences in the overall amplitudes of the waves that were due to differences in head shape and head size and electrode placement and things like that. So that was the strategy. And we found that in our sample, if we aggressively cleaned the ear canal and put the tiptrodes way in, we could get very easily recognizable SP and then APs, and we measured them with 2 independent observers and got the same answers. And so we're going to show you data on wave 1 or AP amplitude, SP amplitude, and the ratio between the two. So to get into the study, the people had to have normal audiograms, but only normal out to 8K. Just as a further little detail here, really the way we defined low risk and high risk was not where they went to school, but how they answered the following question: when you are in a very noisy environment, do you A, always, B, never wear ear protection? And we actually didn't have any trouble finding people who never wore ear protection. We actually did find 12 people who said they always wore hearing protection, and so those were our 2 groups. Admittedly, a very small size, but we figured if we couldn't see anything in this disparate populations, then maybe it wasn't going to be possible to see it at all. So the first thing we noticed was this other kind of hidden hearing loss, which is very easy to reveal because all you have to do is do high-frequency audiometry, but you can see a very striking difference in the high-frequency thresholds between these two groups. And that, to me, as someone who has studied animal models of noise-induced hearing loss for decades, is just screams early noise damage, because in animal models of overexposure, mice, rats, guinea pigs, chinchillas, cats, the literature is full of examples wherever you put a noise band and you raise the level and you ask, where do I first see permanent threshold shift or hair cell damage, the answer is not in the frequency region of the exposure, but the first damage you see is at the very high frequencies. And then only as it gets worse might you start to see threshold shifts in the tonotopically appropriate region. So to me, to us, this was very strong evidence in favor of the idea that this high-risk group was on their way to noise-induced hearing loss as measured by conventional audiometry. And to me, it's very consistent with the idea that these folks really are protecting their ears and these folks really aren't protecting their ears. So here are the speech and noise tests. So we use words in noise, not sentences in noise. We didn't want context effects. We just wanted pure sort of perceptual ability on the single monosyllabic words lists. We used a couple of different signal-to-noise ratios. And as was alluded to earlier, we also used this reverberation test that was actually developed or suggested to me by Doug Brungart as a way to really spread scores. So you speed up the words and you add reverberation. At any rate, you can see absolutely no difference in their word scores in quiet. But for all of the other conditions, the different signal-to-noise ratios and the different speed and reverberation ratios, the difficult tests where even the best people don't do anywhere near 100%, there's a statistically significant difference between the groups. So normal audiograms, statistically significant difference in their word recognition scores, and a difference of a factor of 2 in their SP-AP ratios in the direction that is consistent with primary neuronal degeneration, right? So if the hair cells are okay and the nerve fibers are not, the AP should be smaller, the SP should be unaffected, so the SP to AP ratio should be higher in the folks that are abusing their ears. And very small sample, but that difference is true in both males and females because there are all sorts of reports about gender differences in ABRs. So we and a bunch of other groups are pursuing these questions, trying to get bigger samples, trying to see to what extent you can see electrophysiological signs of wave 1 reduction in normal hearing people that are correlated with problems hearing speech and noise. And obviously we need more tests and much bigger sample sizes. But it's certainly consistent with the idea that these quite young people who are abusing their ears may be producing some kind of synaptopathy. Okay. So to wrap up here, I'm going to talk about the idea, the question, is this, if this really is present in people, and if we can ever figure out how to really diagnose it, is it possible that this might actually be amenable to a therapy? Because, as I said earlier, there seems to be this long, potentially therapeutic time window where the hair cells are all there, and the ganglion cell, and maybe even for a few weeks, the peripheral axon is still there, and you just have to encourage it to send out a new peripheral process. So, how fictional or non-fictional is this basic idea? Well, it's been known for decades that in addition to communicating with each other by neurotransmitters, glutamate, there's a completely different signaling system that hair cells and neurons and supporting cells in the inner ear use that involves ultimately the secretion of this protein called neurotrophin 3, from supporting cells and to some extent from hair cells, that is important for neuronal survival. But even more to the point, 25 years ago or so, a number of groups, most notably the group at Kresge Ann Arbor, showed that if you took a guinea pig cochlea, you could if you destroyed the entire organ of Corti with aminoglycosides and left to their own devices, the ganglion cells would start degenerating. But if instead of leaving it unperturbed after the ototoxic drugs, you perfuse the cochlea with neurotrophins like NT3, not only could you keep the spiral ganglion cells alive, but they showed clear signs that even in the adult ear, these spiral ganglion cells were sending out new peripheral axons. And they were going out to where the organ of Corti used to be and spiraling up and down the cochlea, looking like they were looking for things to connect with. But of course, after aminoglycosides, there's nothing to connect with. So there's just this huge, bizarre array of nerve fibers climbing up and down the cochlea, which don't accomplish very much, although there are people who are excited about using neurotrophins to aid cochlear implants, which is a whole other interesting area. But what's clear is that even in the adult ear, the field of neuroscience, knows enough about neurons in general and spiral ganglion neurons in particular that we know how to activate the signaling pathways that get them to send out new axons. And the idea is, well, what if there's enough information available in the landscape that these new processes can maybe find hair cells and reconnect with them? Then it's not inconceivable that this condition could be reversed. So we've done a number of experiments to ask that question, and we have had a success with a proof of principle experiment where we took mice and exposed the round window membrane and put neurotrophin protein on the round window membrane. So the round window membrane is a membrane that seems to allow some things to pass through it, at least in small quantities. And of course, the round window niche and membrane area are accessible in humans. You could squirt things through the human eardrum and get chemicals onto the round window and do local delivery of drugs. So it's an interesting approach that I think can be translated from animal models to humans. So at any rate, the experiment was a very simple one. We took mice, we measured their ABRs and DPs to get their pre-exposure values, and then a few days later, we overexposed them to the noise band that causes this synaptopathy without any hair cell damage. And then 24 hours after the noise, we opened up, exposed the round window, and squirted this solution of NT3 protein on the round window, and then waited a couple more weeks and looked at the ABRs and DPs, and then cut them up and counted synapses. And there very clearly is something going on. It's very clear that in some of the ears, we were able to reconnect the nerve fibers and the hair cells. So here I'm showing the histology. The black is the synaptic counts as they're supposed to be. Over here we have sham injections that just have the chemical vehicle and a low concentration of neurotrophin in blue, and just zero in on region here, which is where the most damage is, and you can see every single untreated ear or ear only treated with vehicle or low concentration of drug has about the 50% loss of synapses, whereas in the ears treated with the high concentration of neurotrophin-3, some of them didn't look like they worked, but about half of them looked like there was 75% or so recovery of synapses. So apparently, it's possible, at least within 24 hours in the mouse, to encourage some of these neurons to reconnect with inner hair cells. And most importantly, when we measured wave 1 amplitude at high sound pressure levels, down here are the untreated, vehicle-treated, and low concentration treated ears. Here's what it's supposed to be without any trauma. And you can see the ones treated with the high concentration of neurotrophin-3 show partial functional recovery as well. So the same ears that histologically appear to have reconnected functionally appear significantly better than their unsuccessful or untreated other groups. So in conclusion, I've shown you that noise causes rapid synaptic loss and slow cochlear nerve degeneration, even if hair cells recover. There are a couple of studies out there in the literature which say that the same is true of ototoxic drugs. In other words, if you titrate the level of ototoxic drugs down below that, just below that, that starts causing threshold shift, and then you look at synapse counts, that you see synaptopathy in ototoxic ears. So it may be true in both noise and ototoxic ears. I showed you the results suggesting that similar things happen in aging, in mice definitely, possibly in humans, that you lose the neural connections before you lose the hair cells. It's really clear that this kind of diffuse primary neuronal degeneration doesn't elevate audiometric thresholds, and it seems very likely that it must cause deficits in more complex hearing tasks. Briefly alluded to the idea that this may cause tinnitus as well. It's certainly true that noise is the most reliable way to produce tinnitus, that noise exposure doesn't have to cause permanent threshold shift to cause tinnitus. And it's an interesting idea that a number of people are pursuing, that it's precisely the loss of these neural connections that silences a bunch of auditory nerve fibers that then elicits the central compensation mechanisms that lead to the perception of phantom sounds. And for me, the most exciting aspect of it is the idea that the really slow degeneration of the spiral ganglions and its central projections offers a therapeutic window, and it's conceivable that someday we can figure out how to use NT3 or something like it to get axon regrowth and reconnection and actually produce a therapy for this. So finally, let me just acknowledge all of my many collaborators over the years that have been super important for this work. I talked a lot about Sharon. Sharon is my co-discoverer of hidden hearing loss. It was her interest in interactions between noise and aging that caused us to do the experiments in the first place, and we've been working together on it ever since. My wife, Leslie, develops all the immunostaining protocols, and they've really been absolutely key to making all of this work. I talked about Stéphane Maison. He's the one leading the human ECOG study. He's the one who did the one on young people, and he's pursuing it now with a larger sample size. The existence of the Human Temporal Bone Bank at the Mass Eye and Ear is obviously incredibly important to taking this to human autopsy material, and acknowledgments to this talented postdoc who worked with me, and Leslie Shinobu, who was at Decibel Therapeutics, in getting the round window NT3 experiments going. So anyway, with that, thank you for your attention, and I'll answer questions if we still have time.
Thank you, Steve Milo. In one of your later slides where you're showing the improvement with the introduction of the NT3, you showed that some of them reattached, some of them did not. Have you gone into any examination as to why the ones that did not?
The simple answer is we don't know why. The working hypothesis is that it's a drug delivery issue. It's hard to get a big molecule like a protein across the round window membrane, and probably hard to get it through in high enough concentrations to be effective. So we are pursuing different mechanisms of drug delivery, and we're pursuing different assays to be more definitive about which target cells have actually received it. There's a way to do an assay to tell whether a cell has upregulated the downstream players that they should after neurotrophin is delivered to them. So there are a number of tedious tasks that we're currently doing to try to figure out why.
Presumably, some of those synapses that all degenerate at the same time or at the same rate. Is there any study being done on the degree of degeneration as it relates to the level of success of the introduction of the NT3?
There's a lot more detail about who degenerates and who doesn't degenerate, and the rates that I haven't gone into here. That 50% loss that I kept showing you over and over again, it turns out that it's not at all random. The auditory nerve, which I didn't talk about at all, has different subtypes. There's a low threshold subtype and a high threshold subtype, and it's the high threshold subtype that is more vulnerable. They're the ones that go first, which is another reason why the thresholds don't change, because the sensitive fibers remain and the insensitive fibers die. And it's also interesting because we've known for decades that probably the high threshold fibers are really important for hearing and noise because the low threshold fibers are saturated. So it's a clue as to we and a bunch of other people are pursuing what is special about these high threshold fibers that makes them degenerate in the first place. We're probably going to make more progress on that question than which ones regenerate and which ones don't. But I'm pretty sure it's a drug delivery issue.
Thank you.
Going back into your earlier slides, did you vary in your high noise exposure on the variety of your lab animals? Were the bandwidths of that noise exposure different? I thought I remembered seeing different.
Yes, because they were different for complicated reasons. Sharon Kajawa has done a lot more parametric studies of this whole phenomenon, not only changing bandwidth, but sort of trading off intensity and duration. And the phenomenon is not super sensitive to exact parameters. Kind of anything you do, if you find a bandwidth or an intensity duration combination that gets you to the border of TTS and PTS, and you let it recover, and you look at the ear, you will see very striking and significant signs of synaptopathy. I think the biggest unanswered question is, those are very severe temporary threshold shifts. They're a kind that would take a week or so to recover. So what we don't know is how dangerous much less severe TTSs are that maybe take hours to recover or much less time.
The reason I ask is if it was more frequency band sensitive, so to speak, if in the future there was some possibility of having your defenders being specific to, if we got our hands on a particular bandwidth that's much more devastating to the mechanism than others.
Right. Well, in the early stages, it's pretty clear that the synaptopathy is worse in the basal half of the cochlea than it is in the apical half of the cochlea, like a lot of other things that are worse for the high-frequency region than the low-frequency region. But there's sort of an early wave of degeneration that shows that characteristic, and then there's a delayed wave of degeneration that seems more uniformly distributed along the cochlear spiral.
Hi, I was wondering if the acetylcholine enzymes play a role in intercell synaptic transmissions.
I'm sorry, I didn't hear.
I was wondering if the acetylcholine enzymes play a role in inner hair cell transmissions.
Right. So the whole question about cholinergic transmission, which is the transmitter of the efferent system, it's abundantly clear that cholinergic effects are really important in the outer hair cell region. It's much less clear whether they're important in the inner hair cell region directly, but there can be effects via the outer hair cells. And so efferent feedback to the outer hair cells can be protective in these situations and can minimize hair cell damage and synaptopathy. That much is known. It's much less clear if there are direct effects of acetylcholine in the inner hair cell area.
Arthur Boothroyd. I do remember years ago working with profoundly deaf children, there was great fear that by fitting them with high levels of amplification, you were going to destroy what hearing they had, and so monaural fittings became very popular. The levels that are being presented to the ears of the people you're fitting with hearing aids seem remarkably similar to what you were describing as causing synaptopathy, and I wonder if, can you relate the exposure to amplified sound to any of the effects that you're talking about?
Yeah, well, that's obviously a really difficult question because the quantitative details about levels and durations are so species dependent. I think what's abundantly clear is the good news is that humans are less vulnerable in the sense of just absolute sound pressure level. What does it take in a human for intensity and duration to get at the border between TTS and PTS? I think the answer is much more. And it seems to be true in our rhesus experiments as well. So I don't think there's a qualitative difference. I think if you get to that border between TTS and PTS, the likelihood is that the same thing will happen. But I think the good news is in humans it does take a little more sound pressure times duration to achieve that. But then the further caveat to that is that acoustic trauma is complicated and it's many different processes. And a lot of the animal work on acoustic trauma centers around 2-hour exposures because they're convenient to do and to study. And how that extrapolates to much lower pressures for much longer time periods is very difficult to make that extrapolation. That's one of the big challenges in, as you know, in establishing damage risk criteria.
Yeah, I have one. I have the microphone, so I have a question, and then we'll have to move on. But so, how common do you think this is? Do you think that there's, is this like an epidemic of people walking around with faulty auditory nerve fibers, and we're missing them all?
Well, I don't know the answer yet. We're trying to get at that both with the living human study and with the autopsy material study. So we didn't talk about it, but we actually have a way to count peripheral axons in the archival material. So the Mass Eye and Ear has 2,000 human temporal bones in storage. They've only processed 10% of the sections. We can pull archival sections. We can immunostain them, we can count peripheral axons. We've got aging, we've got noise damage, we've got ototoxic drugs, we've got chemotherapeutic drugs. And so over the next 3-4 years, we're going to be amassing data on neural counts in all these populations. And I think that will give us a lot more information about, and counting hair cells too. How widespread is this primary neuronal degeneration based on the real hard empirical facts as to what's really going on in the ear. I think that's the clearest way we'll get the answer.
Thank you. Thanks.